Hello friends, welcome back to my channel MedExam Friendly. If you are new here on this channel, please like, share and subscribe. In this video, we will discuss about antibodies, what is the basic structure of antibodies and the various classes of antibodies and its functions. So antibodies are also known as immunoglobulins. So these are usually produced whenever the antigen or the pathogen it enters in our body. So antibodies are produced in response to the entry of this pathogen. So the characteristic feature of immune system is recognition of antigen by molecules, namely antibodies. So Tiselius and Cabot in 1938 they have shown that antibodies activity was associated with the gamma globulin fraction of the serum. So the term gamma globulin thereafter became synonymous with antibodies. So whenever the human serum is subjected to electrophoresis, it can be separated into different fractions such as albumin, alpha 1 globulin, alpha 2 globulin, beta globulin, and gamma globulin, as you can see in the figure. This is nothing but the electrophoresis of the serum. Whenever the electrophoresis of the serum is done, we get different fractions of albumin, globulin, globulins of various types like alpha 1, alpha 2, beta, and gamma globulins. In 1964, the generic term Ig was internationally accepted for this antibodies. Antibodies are nothing but the glycoprotein molecules that recognizes a particular epitope on an antigen. Usually antigen it will have on its surface, as you can see in the figure, some projections which are nothing but the epitope. So with that epitope only it will bind to particular antibody specifically, and finally it will facilitate the clearance of that antigen. So they are present on the B cell membrane and are secreted by the plasma cells. So antibodies are present in the blood serum, tissue fluid of mammals and are denoted in short as Ig. So whenever the pathogen enters our body, these immune cells they'll get activated, and these B cells differentiate into memory and plasma cells, and these plasma cells it will produce these antibodies. Coming to the structure of immunoglobulins. So all the immunoglobulins will have the same basic molecular structure, and it resembles that of the alphabet Y, and it has 4 polypeptide chains, that is 2 light chains and 2 heavy chains, as you can see in the figure. And it also have a Fab fragment, Fc fragment and Fd fragment of Fd region, which we are going to discuss in the further slides. The basic structure of this antibodies is produced or proposed by this Rodney Potter in 1962. So according to this model, each immunoglobulin it is composed of 4 polypeptide chains, and it resembles an English alphabet Y. So out of the 4 polypeptide chain, 2 smaller chains are called as light chain, and the other 2 longer ones they are nothing but the heavy chain. So these chains, light chains and dark chains, they are held together with the help of disulfide bonds. Coming to the light chains or L chain, so it has a molecular weight of approximately 25,000 daltons, and each light chain is made up of about 210 to 230 amino acids. And light chain and heavy chain, as I have said, it is attached by means of disulfide bond, as you can see in the figure. And there are two types of light chain, that is kappa and lambda. So an immunoglobulin may have either kappa or lambda chain, but never both. And these light chains were named as K or L after the scientists who originally described them. So those are Korngold and Lapre. So after the name of these scientists, these light chains were named as K or L. So L chains are structurally similar in all the cases of immunoglobulins or all the classes of immunoglobulins, be it IgG, IgM, IgE, IgA, IgD. So in all these classes we find L chain structurally similar. Coming to the heavy chain. So H chain has a molecular weight of approximately 50,000 daltons, and each heavy chain it is made up of 420 to 460 amino acids, and two heavy chains they are joined together by means of 1 to 5 disulfide bonds. And there are different classes of immunoglobulins. So these classes they are named after the heavy chain or that is found in that particular class of immunoglobulins, which are designated by the Greek letter as follows, like IgG, in which we find the heavy chain gamma. IgA, heavy chain found is alpha. IgM, mu. IgE, epsilon. IgD, delta. So uh, depending upon the type of heavy chain, these IGs are classified into these five major classes of IG, GAMAD respectively. And the amino terminal portion of the chain, that is nothing but the antigen binding site, helps in binding the antigen, and the part of the antibody that combines with the antigen is called as a paratope. So this binding of antigen and antibody at this particular site, it resembles like that of the lock and key mechanism. So epitope is nothing but the projections which are found on an antigen that will bind to a particular site or specific site present on a particular antibody, leading to antigen antibody reaction. Coming to the Fc fragment. So usually this antibody structure it has two heavy chains, two light chains, and this light chain and heavy chain combinedly form the Fab region, and this Fc region is formed by only heavy chain, and Fd fragment is formed by a part of this heavy chain of the Fab end. So coming to the Fc fragment. So Fc fragment it is composed of carboxy terminal portion of heavy chain, and what it will do is it will determine the biological properties of immunoglobulins, like immunoglobulins usually helps in complement fixation, placental transfer, skin fixation, attachment to phagocytic cell, degranulation of mast cells, and catabolic rate. And this Fc fragment is usually crystallizable. It can be broken down into pieces when they are subjected to particular enzyme like papain and pepsin, and it also contains carbohydrate. Next, coming to the Fab region or Fab end, it is amino terminal half of the heavy chain and one light chain. They form this Fab end, and it does not crystallize, and it does not contain carbohydrate like that of the Fc fragment. Fc fragment it contains carbohydrate, but Fab fragment it does not contain carbohydrate and act as antigen binding fragment. So here only the antigen it will bind with the particular antibody. Next, coming to Fd fragment. As you can see in the figure, Fd fragment it is formed by a portion of this heavy chain. So the portion of H chain present in the Fab fragment is called as Fd piece, and its function is not known. Next, coming to the immunoglobulins function. So immunoglobulins has various functions. Let us see one by one what are the functions performed by this immunoglobulins. The first one is opsonization. So opsonization is nothing but the mechanism of antibody mediated increase in phagocytic efficacies known as opsonization. It usually involved the binding of an opsonin, example antibody, to an epitope on the pathogen, and after opsonin bind to the membrane, these phagocytes they'll get attracted to that particular pathogen and they help in phagocytosis. Next, coming to the neutralization, another function that is neutralization. So this immunoglobulins they help in the neutralization of the toxins and toxoid. Coming to the next function, this immunoglobulin also helps in the activation of this complement system, and the immunoglobulins they prevents the attachment of microbes to the host cell, thus preventing further damage. And they also helps the motility of microorganisms. Restricting in the motility of the microorganisms is also done by this immunoglobulins. And immunoglobulins are usually responsible for this agglutination of the microbes, hence they cannot move and they do not attach to the host cell. So in this way immunoglobulins are helpful in performing the following functions. So the figure given here is that of the agglutination reaction, where the different antigens they are stick together by combining with particular antibodies. Hence they restricts the movement of this antigen in our bodies. Next, coming to the immunoglobulin classes. So we have 5 classes of immunoglobulins: IgG, IgE, IgD, IgM, IgA. You can easily remember it as GAMED, GAMED. So now we are going to discuss in detail about each class of immunoglobulin. So what is the structure, what is molecular weight, what is the function, and what are the subclasses that we are going to learn. So coming to the first class of immunoglobulin, that is IgG. So IgG it is a major serum immunoglobulin and it accounts for about 70 to 80% of total immunoglobulin in the human serum, and it has a molecular weight of around 1,50,000 daltons, and its sedimentation constant is 7s, and it is distributed equally between intravascular and extravascular compartment. So this is the structure of IgG immunoglobulin, that is Y shape, consisting of 2 light chain, 2 heavy chain, and it has a half-life of about 23 days, and it appears to be spindle, and the normal serum concentration of IgG is 5 to 16 mg per ml. Moving on to the functions of this IgG immunoglobulin. So IgG immunoglobulin is the only immunoglobulin that can passes through the placental barrier and provides a natural passive immunity to the newborn. And it also produces passive cutaneous anaphylaxis reaction, and it also participates in immunological reaction like precipitation, complement fixation, neutralization of the toxins and viruses. And it also binds to the microorganisms and enhances their phagocytosis. So these are the functions of IgG. This was about IgG immunoglobulin. Next, coming to the classes of IgG. So there are 4 classes of IgG has been recognized, that is IgG1, IgG2, IgG3, IgG4. So each is having distinct type of gamma chain, as you can see in the figure, so which are identifiable with specific anti-serum. So there are four subclasses of IgG. Moving on to the next immunoglobulin, that is IgA. So IgA is fast moving alpha globulin and it constitutes about 10% of the total serum globulin. Normal serum level is 0.6 to 4.2 mg per ml and it has a half life of about 6 to 8 days. Coming to the molecular weight, it is 1,60,000 daltons and its sedimentation constant is 7s. So as you can see in the figure, this is the structure of IgA. It is usually a dimer having two protein units which are joined together with the help of this J chain or joining chain. Coming to the functions, it is found in high concentration in colostrum, secretions like colostrum, tear, bile, saliva, intestinal and nasal secretions. So the amount of this immunoglobulin is raised in cases of multiple myeloma. Multiple myeloma is nothing but is a cancer of the plasma cells, and this immunoglobulin does not passes through the placenta. And this IgA does not fix complement, but what it will do, it will activate the alternate complement pathway and it promotes the phagocytosis and intracellular killing of the organisms. Next, coming to the subtypes of this IgA immunoglobulin. So it is of two types, that is serum IgA and the secretory IgA. Next, moving on to the next class of immunoglobulin, that is IgM. So IgM, it is a pentamer as you can see in the figure. It has 5 protein subunits which are joined together at the center by the help of this J chain or joining chain. This is the first immunoglobulin that is produced in response to an entry of antigen. So whenever an antigen enters in our body, the first immunoglobulin to be produced in response to that entry of the antigen is IgM, and it constitutes about 5 to 8% of serum immunoglobulins. Coming to its molecular weight, it is around 9 lakh to 10 lakh daltons. So hence it is known as macroglobulin or millennial molecule due to its large molecular weight. And it has a half-life of about 5 days and it is a pentamer as it is composed of 5 monomeric units which are joined together with the help of J chain or joining chain. And each unit of this pentamer has 2 heavy chains and 2 light chains. So in the light chain, it can be either K or L type but cannot be both. And these monomers are arranged in the form of a pinwheel array with the FC end in the center. So the monomers are joined together by short peptide called J chains. And this IgM, it is very rich in carbohydrates and we find it intravascular. Coming to the functions of IgM, so IgM, it participates in agglutination reaction and it helps in phagocytosis and it also inhibits the mobility of the pathogen and it helps in antigen recognition and it is efficient in complement fixation, cytotoxic reaction, etc. Coming to the subclasses, so it has two subclasses, IgM1 and IgM2. Next, coming to IgE. So as you can see in the figure, IgE, it resembles somewhat to that of the IgG and it is reagenic antibodies responsible for immediate hypersensitivity reaction and it has a molecular weight of around 1,90,000 and its sedimentation coefficient is 8s, and it has a half life of about 2 days. So this IgE, it is inactivated when it is heated at 56 degrees Celsius for about 1 hour. It gets inactivated, and it has affinity for surface of tissues cells, particularly mast cells of the same species. So this immunoglobulin also cannot pass through the placenta, and it also will not participate in complement fixation, and it is mostly found intravascularly, and it is found in traces in serum, and we find the elevated level of IgE in atopic conditions like asthma, hay fever, eczema, and also in the children who are having parasitic infestation in the intestine. They also shows the elevated level of this IgE immunoglobulins. Coming to IgD immunoglobulin, it is present in concentration of 0.03 mg per ml, and it is also mostly intravascular, having a half-life of 3 days, and it is present on the surface of unstimulated B lymphocytes, and it acts as recognition receptors for antigens, and it results in either activation and cloning to form antibody or suppression. And there are two subclasses of IgD, that is IgD1 and IgD2. Coming to the figure which is in the extreme corner, it also resembles structurally to that of IgG, IgE immunoglobulins, and very little is known about the function of IgD immunoglobulin. As you can see here, IgG, IgE, IgD, they are monomers and they're structurally similar, whereas IgM, which is a pentamer, and IgA, it is a dimer. Coming to the major differences between all these 5 classes of immunoglobulins. So coming to the structure, IgG, it is a monomer, IgA, it is a dimer, IgM, it is a pentamer, IgD is a monomer, and IgE is also a monomer. Coming to the percentage of total serum, so IgG, it is around 80%, it constitutes about 80% of the total serum, whereas IgA constitutes about 10 to 13% of the total serum. IgM constitutes about 5 to 8% of the total serum. IgD constitutes about 0.2% of the total serum, and IgE 0.002% of total serum. Coming to the location where these immunoglobulins are found. So IgG, it is found in blood, lymph and intestine. IgA, it is found in blood, lymph and B cell surface, whereas IgM is found in secretions. IgD, it is found in B cell surface, blood and lymph. IgE, it is bound to mast cells and basophil cells. Coming to the sedimentation coefficient, IgG has a sedimentation coefficient of 7s, IgA has a sedimentation coefficient of 7s, and IgD also has a sedimentation coefficient of 7s. IgM has a sedimentation coefficient of 19s, and IgE has a sedimentation coefficient of 8s. And coming to the carbohydrate percentage, it is 3% in IgG, 8% in IgA, 12% in IgM, 13% in IgD, and 12% in IgE. Coming to the half-life in days, IgG has a half-life of about 23 days, IgA has a half-life of about 6 to 8 days, IgM has a half-life of about 5 days, and IgD has a half-life of about 2 to 8 days, and IgE half-life of about 1 to 5 days. And the heavy chains which are found in these immunoglobulins are, in IgG we find heavy chains gamma 1, gamma 2, gamma 3, and gamma 4. In IgA, we find heavy chains alpha 1, alpha 2. In IgM, mu. In IgD, delta. IgE, epsilon. And coming to the light chain, so all the immunoglobulins, they either have kappa or lambda chain, but never the both. And complement binding, the immunoglobulins that helps in complement binding or fixation is IgG, IgA and IgM, whereas IgD and IgE, they do not participate in complement fixation. Coming to the placental transport, so IgG, it is the only immunoglobulin that can pass through the placental barrier and provides the natural passive immunity to the newborn. And then immunoglobulins that are present in milk are IgG, IgA, and the immunoglobulins that are found in the seromucous secretions are IgA. And coming to the heat stability, IgG, IgA, IgM, IgD, they all are heat stable except for IgE. So thank you. If you like my video, please don't forget to like, subscribe and hit the bell icon for further notification.